Welcome to Physio Exam Buddy. This video deals with the erythropoiesis, mainly the signs as well as the stages of erythropoiesis. We all know that erythropoiesis means it is the entire process by which RBCs are produced, and it starts in the third week of intrauterine life. Next is about the sites where erythropoiesis occurs. It differs during the intrauterine life as well as the extrauterine life. First, we will see about the intrauterine life. We can divide the intrauterine life into three phases based on the site at which erythropoiesis occurs. The first one is the mesoblastic phase, second one is the hepatic phase, and the third one is the myeloid phase. During the intrauterine life, we already discussed that the erythropoiesis starts from the third week of the intrauterine life. So the first phase, that is starting from the third week to third month of intrauterine life, erythropoiesis occurs in the mesoderm of the yolk sac. So this phase, it is known as the mesoblastic phase, and it is also known as a stage of intravascular erythropoiesis, followed by the mesoblastic phase starting from the third to fifth month of the intrauterine life. Erythropoiesis occurs in the liver, spleen as well as in the lymph nodes. So this phase, it is referred to as the hepatic phase. And following the hepatic phase, next phase is the myeloid phase, that is from the fifth month till birth of the baby, and the bone marrow will be producing the RBCs during the myeloid phase, especially the red bone marrow. During the extrauterine life or after birth, red bone marrow is the main site of production of red blood cells. Up to 6 years of age, all bones in the human body have red bone marrow. So, the RBC production or the erythropoiesis occurs in almost all the bones up to 6 years of age. But from 6 to 20 years of age, erythropoiesis occurs only in the long bones as well as in all the membranous or flat bones. And the reason is that in other bones, the bone marrow is replaced by the yellow bone marrow, and we all know that the site of production is only the red bone marrow. So the red bone marrow is seen only in these long bones as well as in the membranous or flat bones. That is why RBC production between 6 to 20 years is happening only in these bones. After 20 years, erythropoiesis occurs in the ends of long bones like humerus and femur, as well as in the membranous bones like cranial bones, ribs, sternum, vertebra, and pelvic bones. And as age advances, this red bone marrow becomes less productive. And whenever there occurs any increased necessity to the body, this yellow bone marrow is getting converted into red bone marrow and will begin to produce the red blood cells. And if there is still more necessity which can't be managed by this yellow bone marrow as well as the red bone marrow, the liver and spleen will start producing the red blood cells. So, whenever the liver and spleen starts erythropoiesis, that erythropoiesis, it is known as the extramedullary erythropoiesis. Next, we will see the stages of the erythropoiesis. Almost all blood cells, that is red blood cells, WBCs as well as the platelets, it originates from a pluripotent hemopoietic stem cell. From this pluripotent cell arises the committed stem cell, and committed means it is committed to produce only one series of cells. So here in case of erythropoiesis, pluripotent hemopoietic stem cell give rise to committed stem cell mainly of the myeloid series, and from there it is converted it into burst forming unit erythrocyte, and from burst forming erythrocyte, the colony forming unit erythrocyte is formed. And from the colony forming erythrocyte gives rise to proerythroblast, from then into basophil erythroblast, then to polychromatophil erythroblast, next orthochromatic erythroblast, which is converted it into reticulocyte, and finally it will get mature into a erythrocytes. So this is in short the stages of the erythropoiesis. Now we will see in detail about each stage of this erythropoiesis. Pluripotent hemopoietic stem cell. This is one of the most primitive cells. As the name suggests, pluripotent. It gives rise to all types of blood cells. This pluripotent hematopoietic stem cell got two properties. First one is self-renewal and the second one is differentiation. Self-renewal means it is the ability to produce new stem cell. Differentiation means ability to differentiate into mature specialized blood cell. So these two properties are present for this pluripotent hemopoietic stem cell. And from the pluripotent hemopoietic stem cell arises the committed stem cells. This committed stem cells means it gives rise to only one group of cells. So there are two types of committed stem cells in the human body. The first one is colony forming unit spleen or committed myeloid stem cell. And the second one is committed lymphoid stem cell. And this committed stem cell then give rise to progenitor cells. And will be separate pools of progenitor cells for different blood cells. And the progenitor cells for erythroid series, that is for the erythropoiesis, is mainly the burst forming unit erythrocyte as well as the colony forming unit erythrocyte. So this is the flowchart showing the erythropoiesis as well as leukopoiesis as well as thrombopoiesis. So we will now concentrate mainly on the erythropoiesis. So all cells, it arises from the pluripotent hemopoietic stem cell which give rise to the committed stem cell of myeloid series. Another one is the lymphoid series which give rise to the T lymphocytes as B lymphocytes by leukopoiesis. But now we want only committed stem cell myeloid series which gives rise to burst forming unit erythrocyte and next into the colony forming unit erythrocyte, and from this the erythrocyte arises. Whereas here this part is mainly the leukopoiesis as well as the thrombopoiesis. So in this video we will concentrate mainly on this portion, that is the erythropoiesis. So the progenitor cells, that is the committed stem cell myeloid series, give rise to the burst forming unit erythrocyte which will differentiate into colony forming erythrocyte in around 6 to 7 days. And the conversion of BFUE to CFUE requires certain growth factors like interleukin 3 and granulocyte monocyte colony stimulating factor. And the late stages in the conversion of the BFUE to CFUE, they are dependent on the hormone erythropoietin. Next is the colony forming unit erythrocyte. Colony forming unit erythrocyte give rise to the pronormoblast or the proerythroblast. Again, this conversion, this stage is also erythropoietin dependent. Next is about the pronormoblast or proerythroblast. So this is the first recognizable erythrocyte precursor cell and they have a diameter of around 14 to 19 micrometer. So whenever you are drawing the erythropoiesis stages in your exam, this is the largest cell to be drawn, that is 14 to 19 micrometer. And whenever the stage progresses, the size should decrease it goes on decrease in the size. Next is about the nucleus. You can see here that the nucleus is very large one, occupying almost 8% of the cell and it contains nucleoli. So when you are drawing the nucleus, you have to shade it using hematoxylin pencil itself. Again, the nucleoli you can color it using light blue color pencil. And the cytoplasm is actually a rim of basophilic cytoplasm, which again you have to color using light blue color pencil only. And another important feature of the pronormoblast or the proerythroblast is that ferritin molecules are present inside this pronormoblast. And this pronormoblast or proerythroblast is next converted it into the next stage, that is the basophilic normoblast or erythroblast or early normoblast. You can see here that the diameter here actually decreases to 12 to 17 micrometer. That is, as the stage advances, you have to decrease the size of the cells when drawing the picture of erythropoiesis. Now we will see the nucleus. Nucleus, what happens to the nucleus? The nucleoli is disappeared when compared to the previous stage. Also, the size of the nucleus should be decreased. So, while drawing the basophilic normoblast, the size of the nucleus should be decreased than the previous stage. And you can also see a wheel spoke arrangement, also no nuclei. Here also the nucleus should be shaded with the hematoxylin pencil. And there is a cytoplasm around the nucleus which is deeply basophilic, and it is mainly because of the increased amount of the ribosomes. That also you have to shade it using light blue color pencil. And the condensation of the chromatin, that is the formation of the heterochromatin, begins at this basophilic normoblastic or early normoblastic phase. That is one of the importance of this stage of erythropoiesis. And from the basophilic erythroblast arises the intermediate or polychromatophilic normoblast or erythroblast. Again, the size should decrease when compared to that of the previous stage. Here, the diameter is around 12 to 15 micrometers. Again, what happens to the size of the nucleus? It becomes smaller, and also there is no nucleoli, and there is an increase in the condensation of the chromatin. So you have to color it using dark hematoxylin. Now, what happens to the cytoplasm? It should be colored both blue as well as pink. That is because of the appearance of a faint blush of hemoglobin near the nucleus. So, you have to actually first shade the cytoplasm using light blue color pencil, and you have to mix it with red color pencil around the nucleus because you have to show the appearance of the hemoglobin at this space. So another MCQ you can expect from this portion is that appearance of hemoglobin occurs in which stage of erythropoiesis? So the answer will be intermediate or polychromatophilic normoblast or erythroblast. And another feature in the cytoplasm is that the number of mitochondria, it is found to decrease in this polychromatophilic normoblastic phase. And from the polychromatophilic phase arises the orthochromatic or the late normoblast or late erythroblast. And still the diameter is found to be decreased as the stage advances. So here the diameter, it is only 8 to 12 micrometers, and it is the smallest of the nucleated precursor. Means after this stage the nucleus disappears. So this is the last stage at which you have to draw the nucleus. So you have to draw the nucleus using blue color pencil, that is the hematoxylin pencil, and the cytoplasm using red color pencil, because the light blue color have to be replaced by red color all around the cytoplasm, which shows the appearance of the hemoglobin. So here, after some time, the nucleus will undergo pycnotic degeneration, and it will be finally extruded out of the cell. So this one is the smallest of the nucleated precursor, and after this stage the nucleus will be disappeared. And cytoplasm will be same as that of the mature RBC, that is using red color pencil you have to shade the cytoplasm. And after this stage comes the reticulocyte stage. So here the diameter is just more than that of the red blood cells. Are you seeing any nucleus here? There is no nucleus because it is already extruded out after the previous stage. And what you are seeing in the cytoplasm here, the early stage in the cytoplasm, it contains ribosomes, mitochondria, as well as Golgi complex. But as the maturation occurs, all these organelles will disappear, and mitochondria is the one which disappears first, and the ribosome is the one which disappears last. So how will you draw a reticulocyte? It should be same diameter or slightly more than that of the red blood cells, and you should not draw any nucleus in this reticulocyte, but instead of this you have to draw certain reticulum thread like structures using blue ball point pens. You have to put small dot like strings, demonstrating the reticulum, and the cytoplasm is again red color because of the presence of the hemoglobin. And this reticulocyte, how is it demonstrated? This is demonstrated mainly using the supravital staining, and the stain used is mainly the brilliant crescent blue. And whenever we stain the cell using this brilliant crescent blue, there occurs precipitation of the ribosomal RNA, which will appear as the narrow strands traversing the cell, which is our reticulum, which you have to draw using the blue ball point pen. Small dot like strands you have to draw in the cytoplasm of the reticulocyte. And you should also know the normal count of the reticulocyte, that is, it is mainly 0.5 to 1% of the RBC count in the adults, or 2 to 6% of the RBC count. So for knowing the reticulocyte count, you should first know the RBC count, then only you will get the reticulocyte count. So in case of a normal adult, it will be 0.5 to 1% of the RBC count, whereas in case of infants, the reticulocyte count will be equal to 2 to 6% of the RBC count. And from the reticulocyte arises our mature red blood cells, or the erythrocyte. And this is the last diagram you have to draw in the stages of erythropoiesis. So how will you draw a RBC? The RBC, it is mainly decreased in size when compared to that of the previous cell, and again the cytoplasm is red, is shaded using the red color pencil. But what is to be there in the center? There should be a central pallor, and the central area of the pallor, it is usually about 1/3 of the cell's diameter. Okay, so the pallor should be only 1/3 of the cell's diameter. That is how you will draw a normal erythrocyte. And what are the changes you are expecting during erythropoiesis? There occurs a reduction in the size of the cell. That is, as the cell progresses, there occurs a reduction in the diameter of the cell, and there will be disappearance of the nucleoli as well as the nucleus, and there occurs the appearance of the hemoglobin, and there occurs the changes in the staining properties of the cytoplasm. So this is in short the changes occurring during erythropoiesis. Thank you.